Oh, any question here? Okay, so your question was like, why we have many EPR?
Yes.
molecules? So that's what usually the receptors are formed. Receptors are commonly called many protein coming together. So that's the way it is.
The receptor for glucosulfate, is it outside circular or inside?
So where is the membrane?
Membrane, right?
Membrane should be, yeah, so the receptor should be,
It's right above the afterzone.
it should be like this, right? If this is the membrane, should be like this, right? Because if it is inside the membrane, then how they
Yeah,
connect?
yeah, yeah.
So membranes are always like transmembrane proteins. Receptors are always transmembrane proteins. Okay, now, so that was the fast block to polyspermy. And now we are talking about the slow block to polyspermy. Now the change in the membrane potential, that is transient. That does not stay for a longer time. So membrane potential change is transient. Lasts for a minute or so. Just lasts for a minute. So if that is true, then if we have a condition like this, can you see all the sperms attached to the egg? Right? So there are so many sperm attached to the egg, how they will be removed. So this is so for that purpose, the second mechanism is activated to prevent polyspermy permanently. So that can be transient, right? Some will, but some are still coming. They're still moving, right? So to prevent polyspermy permanently, another mechanism is activated, and that mechanism is called slow block to polyspermy. So idea here is, what is the purpose? Permanently means you want to remove all those sperms which are attached to white line envelope, right? So you want to remove those. For that, you need to have a mechanism which can permanently remove these sperm from the egg. That doesn't allow those sperm to reach to the egg membrane. And for that, this slow block to polyspermy is initiated, and that slow block to polyspermy is caused by cortical granule reaction. That is caused by cortical granule reaction. And when that cortical granule reaction occurs, cortical granule reaction creates a fertilization envelope. I'll show you what that is. Envelope around the fertilized egg. Now egg is fertilized, right? One sperm has entered, so egg is fertilized. So you need to create some barrier. Let me show you the video first. So if you look here, so once the first sperm enters, right? This is the egg, and observe what is happening here, right? Just observe it. So once the sperm entered, you saw that something originated from here and that surrounded it. That is called fertilization envelope. Okay? So that fertilization envelope is created. And how that is created? So we know that so right here is the egg. This is the egg membrane. Right? And outside the egg membrane, you remember we said this is the white line envelope. This is your white line. Or in the case of the mammal, you call it zona pellucida, right? Now this mechanism is active both in mammals as well as sea urchin. The slow block. The slow block of polyspermy is not present in the mammals, okay? That is present in sea urchin and other sea animals, other organisms. But slow block is present in humans as well, okay? So what happened? So remember that beneath the egg membrane, we had these structures. They are through. What you call them? What are these? Cortical granules. So they are around, so these cortical granules are there. Almost 1500 we said they are present in the sea urchin egg. Okay. Now what happens? Say for example, this is the place from where the sperm has entered in the egg. The fusion has taken place, right here. So where the fusion is going to take place, at that point, this particular point, what is going to happen? This process will start, the cortical granule will start at the point of entry of the sperm, right? So right here, what is going to happen? This cortical granule will move and basically fuse with the membrane of the egg. This is after the sperm has fused with the egg membrane. Okay? Now you don't want any more sperm to enter there. So this cortical granule is going to fuse there. And what cortical granule is going to do, it's going to throw all its content right here in the space between white line envelope and the egg membrane. Now, so if we remember what was there in the cortical granule, we said there were proteases, right? So there are number one thing present, serine proteases. Proteases mean that they digest all the proteins. Serine means their target is the serine amino acid. Wherever the serine amino acid is, they're going to go and chop that out. And so it means here they are releasing proteases. Remember what we said the other day when we were talking about this structure, we said that this white line envelope is secreted by the egg, and then white line envelope is attached to the egg membrane by these bridges. Right? We said these are the glycoprotein hosts which keep it attached to the egg membrane. So now what happened? Now you have the serine proteases here. So serine proteases are going to basically kind of digest these glycoprotein hosts, right? Because they are proteases, so these glycoprotein hosts are no more there. So they are gone, they are away, right? Because the proteases have digested. Making sense? So now this is not there. Then what happened? The component of the cortical granule, what they are going to do, they are going to kind of coat, so all those things are coming out, they're going to coat the white line envelope. They start coating here. Okay. This coating which they are doing, that coating is called, and when they coat it, we call it, they create a fertilization. They create a fertilization envelope. Okay? Now what happened? Right here, they were also having some mucopolysaccharides. So remember that we said they also have some mucopolysaccharides. So here, you also have mucopolysaccharides are present in the, they are part of the contents of cortical granules. Okay? Now mucopolysaccharide are sugar. These sugar are exactly like the sugar you make the jello, right? So what happens? You get jello, little bit powder, you add water and that swells, right? So same way it happened that these mucopolysaccharides which are secreted here, they absorb water, and swell. And what that does, when the water is here, now those bridges are broken, this rump is formed. What is going to happen if this is going to swell? This is going to form a structure like this, right? Making sense or no? Right? So now, this process that has started here, that spread through this. Now, this cortical granule come here, this cortical granule come here, this come here, this come here, and ultimately it surrounds the whole pack. In this way, this fertilization envelope is created. Right? Now, this process, the first cortical granule comes here and cause exocytosis, start the formation of fertilization envelope. That happens 20 seconds after sperm egg. That's why we call it late. That start 27 after, and it is completed in a minute. And it is completed in a minute. Now, so the other thing happened there. Now this envelope is formed. You need to stabilize that envelope, right? So what happened? These cortical granules which are there, they have, so whatever content there is, we are naming many things here. They also have contain enzyme called UDX1. Okay? And they also have, so they have UDX1, and then they also have ovoperoxidase. That is the other thing. So number one, number two, that's the second thing. And the third thing, they have these what we call transglutaminase. Okay, now what happened? These three things are there. UDX1 it utilizes oxygen and produce hydrogen peroxide. Okay? So when that is produced, ovoperoxidase and transglutaminase, they utilize H2O2. So this H2O2 which is made by UDX1, right? And what they do, they cross-link adjacent proteins and stabilize fertilization enzyme. Okay. Now, then the cortical granule, cortical granule also secrete hyaline proteins, and what they do, they coat the fertilized egg. So in this way, what is happening, if you think about it, again, we are saying they are releasing serine proteases. Okay? Serine proteases are coming out, they are breaking up these glycoprotein bridges which were joining the white line envelope with the egg membrane. That's the first step. So it can now expand. That connection is gone. Then the material which was in the cortical granule, that kind of coat the white line envelope and make a fertilization envelope. Then you have mucopolysaccharides here. Those mucopolysaccharides absorb water and swells, so this thing goes up. Now this thing starts here, it's spread throughout. So starting here, but here, everywhere. And then what happens? There are enzymes in this UDX1 that utilize oxygen, make hydrogen peroxide. Then you have ovoperoxidase and transglutaminase there. What they do, they utilize hydrogen peroxide and basically cross-link the proteins and stabilize the fertilization envelope. Then from the cortical granule, there is a hyaline protein. That hyaline protein is now going to coat the egg. So in this way, the complete block to polyspermy is going to take place. So what is going to happen in this process? So in this process, what is going to happen? We are going to have a situation where this envelope start forming here, and when this envelope is forming, essentially you are removing the white line envelope from there, and all the chromosomes which are attached there, they are kind of go there. So in this way, the polyspermy is blocked. Making sense? Okay, talk to each other for a minute, then we'll go further.
Serine proteins and mucopolysaccharides, the enzyme UDX1, the ovoperoxidase, the transglutaminase, and the hyaline protein.
So serine, that's all.
Yeah, I think they're all from the cortical granule.
She just have it spelled. That's what she was asking.
Ah, okay.
And then it starts, all of this starts, I mean, start cutting the mammalian envelope.
Say it again. Portal drainage
This happens in
release
30
the fluid. What is the purpose of that?
Enter is gonna take time.
To break these bridges.
UDX will be
Right.
opposite.
And these bridges are there because purpose here is to move away from the Whiteland River. So Whiteland River is attached to the egg membrane through these bridges. So if these bridges are moved, only then you can remove the water.
So we have the serum.
Yeah.
So there's no harm to that.
No, no, no. In the end, I was just kind of repeating what I said in the
Okay.
beginning. Yeah. Yeah. So silk protein is present in every part. So it will start from here, then it will go.
And this.
I feel till like 2 in the morning,
Make sure it's a-
because I started writing it.
Yeah, everything is there. Everything is there. All over. That's what when we are talking about when we're explaining the egg structure, then we talk about it. And we told these things are present there, right? So now we are telling what actually we are. Any question here? You guys are okay? Okay, now. So what initiate this process of cortical granule reaction? Okay? So calcium ion, they are the initiator. They are the initiator of cortical granule reaction. And you can imagine like calcium ion are doing so many things. They are the most critical ion, signaling ion or second messenger in the cell. So upon fertilization, what happens? As the fertilization takes place, upon fertilization, at the site of sperm entry, immediately after the sperm enter, the calcium ion concentration increases. Calcium ion concentration goes up. And when the calcium ion concentration goes up, what is going to happen? That is going to cause the exocytosis, right? Exocytosis is controlled by the calcium. So it means the calcium ion at the site of sperm entry goes up as the sperm fuses with egg. And when that happens, that leads to exocytosis. And essentially, when we say exocytosis, it's essentially membrane fusion. So membrane of cortical granule vesicle fuses with the egg membrane, and that causes the release. Now, we can actually observe this calcium under the microscope. So there are certain dyes there's one dye called Fura 2A. If you take this dye and you put that dye in the media in which egg is growing. So when the calcium binds with this dye actually flourishes, gives a fluorescence. And you can actually track the calcium. So if they do this thing, I can just show you what happens there. Here. So this is the egg. So this is the point of entry of the sperm. Right at this you can see high fluorescence. As the time is, this is the other egg. This little late, this sperm enter there, and you can see it's flourishing. As the time enter, you can see this increasing calcium start here. It is spreading across this, then it's going to the other end. That's why we said that wave of cortical granulation start from here, that spread. Okay? Same way, this is the other egg, you see, this is here, it come here, and it basically going to the other direction. Now let me show you this video, which is corresponding. So first calcium entry take place, right? And to follow that, you have the involved formation take place. So here is the video that we show you. Okay, so these are two videos. The right one is showing you this one is going to show you the calcium, okay? Look here. And this is going to show you the envelope formation, okay? So again, what they have this egg. This egg is now in a media. That media has Fura-2 dye. So when the calcium is going to go up, the dye is going to shine. And you see what happened there, and then we can follow after that what is happening here. There's some sound also. Now you can see here that this wave has now reached here. It started from here where the sperm entered, and once this wave passed, you start seeing the formation of fertilization envelope, right? So that means that the fertilization envelope precedes the calcium wave, right? So first the calcium comes. So now, if you think, okay, the calcium is the real initiator, so from where calcium comes? So calcium in this case comes from within the egg. It doesn't come from outside, okay? So calcium comes from within the egg. And if you take a ionophore, so ionophores are basically chemicals which allow ions to pass through. And there's special called A23187. So this ionophore, what it does, it is basically a calcium ionophore. It's specifically for the calcium. It's a calcium ionophore. And what it does, it releases calcium from all the storages inside the cell. So if you take an egg, you throw this thing there, and what you will find immediately cortical granule reaction will start without any sperm. No sperm entry, just adding this ionophore, release the calcium, and you have the cortical granule reaction start. So that clearly shows, people have done this experiment, that calcium is the key regulator here. So now, what happened that we are saying that calcium is released, all those things released, then what is going to happen there? Then what happened is this whole process of cortical granule reaction and the other things which are going to happen. So something has started, so think about it. Egg was formed and egg was laying there about to die, right? Sperm entered, immediately calcium burst occurred, cortical granule reaction occurred. What does it mean? It means that the egg metabolism which was dormant, that has been activated, okay? So that means what has happened is activation of eggs metabolism. Because that is going to start the development, right? So when you talk about the fertilization, so fertilization is not, this is important, is not merely the fusion of haploid nuclei. This has actually no meaning in fertilization. Fertilization, actually, what is the role of fertilization? Fertilization means sperm entry. It is not, I'm not talking about like the diploid nuclear formation, the sperm entry. What it does, fertilization actually initiates the development process. It initiates the development process. And that development process, formation of new organism, does not involve fusion of haploid nuclei. So haploid nuclei, now we will say what I'm talking about. You know, on one side I'm saying that the diploid nuclei is required, right? I'm saying development is starting even without the participation of the nuclei there. So there are actually examples. In a species, what we call silver salamander, silver salamander. Silver salamander, this species consists only females. There are only females in this species, no male. Okay? What this species does, it lays diploid eggs. It lays diploid eggs. Now, diploid egg is there. You will say, hey, if the purpose of fertilization, that's why I put there. If the purpose of fertilization is having n plus n making 2n, then this diploid egg, which is already 2n, it should just start developing, right? Because it has the chromosome number, just lay egg and start the formation of new organism. But it never happens. So what happened that this species, which is silver salamander, this species actually mates with Jefferson salamander. It mates with Jefferson salamander. Now, Jefferson salamander, the sperm come, attach here, cause the fertilization process, and sperm degenerate. So what sperm does? Sperm only stimulate eggs metabolism, does not provide nucleus. Nothing. So what happened? So it means egg has all the component to make a new individual. But there was some inhibition on that start of the process of the formation of the new individual, right? So there was some inhibition of initiation of development of new organism. And that inhibition is removed by fertilization. That is making sense? So it has nothing to do with the nuclear combination or nuclear recombination. So then what causes it? What made this inhibition go away? Throughout the animal kingdom, we talk about any species, calcium ions are the initiator. Calcium ions are used to start the development process. So that means calcium ions, they are doing two things here. Number one, they are providing block to polyspermy. Number two, they are initiating the egg's metabolism and inhibiting the halt on development process. So whatever halt was there which was blocking the development, they are inhibiting that halt. So they are kind of activating the egg metabolism and saying, hey, now you start the development of new organism. That is making sense so far? Right? Yes or no? You guys are okay here, right? Okay, so then, if that is, calcium is so important, then from where the calcium is coming? And how the calcium is coming, right? So, release of intracellular calcium. So, if the release takes place, then what happens? What calcium is actually doing here? How it is working? There are two views about this. There are two views. People have different views. So one view is that the sperm binding to egg, and that binding was binding with receptor, and receptors were ABR1, right? They say that binding somehow initiate a cascade, and that cascade somehow release calcium ion. The other view is that there are some soluble factors in the sperm. So they are saying it's not binding, but something when the sperm content enter in the egg, there are some soluble factors in the sperm. And what they do, they are saying they initiate calcium release. So two views are there, right? But whatever view is there, whatever, either this or that, and we'll talk about these views in a minute, what can happen. Either this or that, inositol triphosphate, IP3, is the key. So if you take an egg and you inject IP3, this is actually now becoming more interesting. So you take an egg, you inject IP3 into that. Cortical granule reaction will take place, egg will start development. Even there is no fertilization. Now if a fertilized egg is there, there are inhibitor of IP3, we know they are chemical inhibitor. You put that chemical inhibitor in the egg, even the fertilization has taken place, development is not going to take place. So that means IP3 is the key molecule.
Do they both think that? Like
Say it again.
Both paths think that IP3 is the key, right? That,
IP3 and?
and inositol triphosphate. Wait,
Like both,
I'm confused. Like both paths
views.
think that it's.
So whatever the way is, the calcium will be released by IP3. Okay? So IP3 is the initiator of calcium. So that's why they're saying whatever happens doesn't matter. If you inhibit IP3 from functioning, development will not take place. If you just inject IP3 from outside, development will start. So that means IP3 is the key. So IP3, that means is downstream of those things, right? So now what happened, how this IP3 is playing role there? So this is your membrane of the egg. So egg membrane is there. In the egg membrane, you have, we did talk about this, we said there is phospholipase C. Now this phospholipase C, which is present here, this is actually called gamma PLC. gamma phospholipase C is there. And then there is, right here is a so this is the structure of this molecule, what we call PIP2. Phosphorinositol biphosphate. This is right here. So this is actually a lipid anchored carbohydrate. So this portion is a carbohydrate, this portion is a lipid. Okay? Now, what they are saying, what is happening there, when phospholipase C is activated, okay? When phospholipase C is activated, what it does is work on this PIP2. So phospholipase C activation is activation, work on PIP2. And what it does, it produces two things right here. So this portion is released here, and this becomes IP3, inositol triphosphate. It goes into the cytoplasm. Okay. Then the lipid portion here, that stays here, that is right here, that is called DAG. DAG means diacyl glycerol. Diacyl glycerol, DAG, it stays there. Diacyl glycerol stayed there, DAG stayed there. Now what happened? This IP3 is going to, this is the rough endoplasmic reticulum of the egg. Right here on this, there are, right here is IP3 receptor. So IP3 coming here and bind to IP3 receptor. And in the cell, endoplasmic reticulum is the storage of calcium. So all the calcium ion are there. So say this is all these are the calcium ion. So all these are the calcium ion there in the ear. So once IP3 bind here, that open the IP3 channels. So IP3 receptor, there are IP3 channels to which, and that what that does is that cause release of calcium. So calcium is now in the cytoplasm. Okay. On the ER itself, there are another channel right here. So this is called calcium activated calcium channel. Calcium activated calcium channel. And when those are there, so this calcium now, this is coming out from here, it come here and bind to the calcium activated, some of this bind here to the calcium activated calcium channels. And that open these channels throughout, and that release more calcium is coming out. More calcium. So now this cytosol is kind of loaded with calcium ion. So there are a lot of calcium ion there. Okay. Now what happened? This DAG, which was part of this reaction, this DAG actually activates protein kinase C. It activates protein kinase C. So protein kinase C is activated here. Now, when this protein kinase C is activated, along with huge amount of calcium, they both are there, what they do, right here are, this is your protein, proton, sodium ion, antiproton. So both activated protein kinase, that goes here, calcium also goes here, these together, they open these protein, proton, sodium antiproton. And what that does, that essentially hydrogen ions start coming out, and from here, the sodium ion getting there. And when that happens, that increase alkalinity of eggs cytoplasm. So egg become more alkaline, more basic. And what that does, once that thing happens, that stimulates DNA synthesis. That stimulates RNA synthesis. And that also stimulates cell division. And when we say cell division, it means the nuclear envelope of the egg and sperm should start breaking down. So essentially, membrane, nuclear membrane breakdown. Now, if experimentally, you don't worry about IP3, don't worry about calcium. Somehow you increase the alkalinity of the cell cytoplasm, the cell division and the development is going to start. So what is happening there? Let me go back. So we are saying two views are there for the calcium activation. One view is that when the binding binds the receptor on the egg, that stimulate the release of the calcium. The other view is that there are some soluble factors, and those soluble factors are coming with the sperm. They stimulate the cascade of events, and that causes release of calcium. In both ways, whether it is through the binding receptor or through the soluble factor, the calcium release is controlled by inositol triphosphate. Inositol triphosphate is produced by activation of phospholipase C gamma. It's a gamma form of phospholipase C which is in the egg. And once that is activated, that basically work on phosphatidylinositol biphosphate, PIP2, and basically chop it into two parts. One is IP3. IP3 comes to rough endoplasmic reticulum, bind to IP3 receptors, which are on the IP3 channels, and open those channels. Once those channels are open, calcium comes out. This increases the concentration of calcium, but some of this calcium come here and bind to calcium-activated calcium channel, which are on the ER itself. Bind there and open them. Now your cytosol is flooded with calcium ion. Huge increase in the calcium. And then, in the meantime, DAG activates protein kinase C. Both activated protein kinase C and calcium together work on sodium proton antiproton and open it. That causes influx of sodium ion inside the cytosol that increases the alkalinity of the cytosol. So that increase in alkalinity of the cytosol immediately stimulates DNA synthesis, RNA synthesis, and not only that, actually amino translation also. And cell membrane breakdown, cell division start, So essentially, the development starts right here. That is making sense? So if you take an egg and you don't do any of this thing, you just increase the alkalinity of the cell or the egg, without fertilization, it will start, initiate the development process. That is making sense? Okay, so talk to each other for this, then we'll go further.
Yeah, they agree.
Yeah, also like this. And that, and that's activated or causes for activation of GIP2, which then causes IP3 to go to
Which one's the lipid one? You said the lipid one. Did you go through it? It's like a lipid something. What is it?
I heard that.
So IP3 is not often IgG receptor or that lets calcium, or that would calcium channels, and then the calcium channel, or the calcium goes back and binds to calcium activated calcium channels, which allows more flow, more calcium out. And then the PIP2 also became DG, activates the DPNC, which opens the C tap for the sodium hydrogen channel with the calcium, which then increases the flow of sodium in, which increases the alkalinity of the egg and cytoplasm, and then that stimulates egg synthesis, DNA synthesis, cell division, mRNA translation, and then the development of brain breakdown.
Yes.
What did you say about the PIP2, like
It was like a lipid
lipids?
So EIP2 is a lipid anchored carbohydrate. So carbohydrate portion is actually IP3, and lipid portion is this DAG. Okay.
Yeah, I think I use the carbohydrate.
Everything is okay?
Lipid anchored carbohydrate.
You are okay here? You guys are fine? Okay, then. So what happens actually when people start looking at, okay, what is happening when the sperm fuses with the egg, right? So what they find out, that when you have this sperm and egg fusion, that is this binding with the receptor. Okay. Once that happens, that leads to, so remember, if you remember from your cell biology, we said there are so many G proteins which are attached to the receptor, right? So that lead to activation of heterotrimeric G proteins. That lead to activation of heterotrimeric G protein. Okay, so sperm binding to the egg membrane lead to activation of heterotrimeric G protein. And once that happens, that activate a Src kinase. Src kinase, these are the kinases which are involved. The name Src come from sarcoma. So initially they were discovered in the cancer, that's why they call them Src kinase. And the special kinase which is activated here is tyrosine kinase. Tyrosine kinase. That type of Src kinase is activated here. And once that Src kinase activates there, this Src kinase activates PLC gamma. So we were talking about the PLC gamma. So PLC gamma is activated by Src kinase, and that Src kinase is activated by heterotrimeric G protein activation, and that is activated by binding of receptor to the binding protein. Okay. Once this is activated, so we know that we have this PIP2. This PIP2 changes to IP3 plus DAG, right? So this DAG activate protein kinase C, and this IP3 leads to calcium activation, and these together, PKC and they together activate sodium proton antipoda. Now, that is one thing is happening. But this increase in calcium, it does one more thing. It starts cortical granule reaction. That is slow block to chlorisperm. So these are happening side by side, right? On one side you are blocking the entry of other sperm. Now, this activation channel, what it does, increase and that what it does is essentially inactivate MAP kinase, and that starts cell cycle. So, this is if the sperm is binding to the, if this cascade is activated by binding to the receptor, right? And if there is soluble factor, you can say if the soluble factor is there, then these things are not going to happen. So soluble factor, because soluble factor is directly entering in the sperm, right? From the sperm to the egg. That soluble factor is going to activate the PLC gamma. Now, what is that soluble factor? We'll talk in the next lecture, right? So, idea is that we have to go to the cell signaling. When the sperm is binding, that is happening at the cell membrane. And we know that the membrane signaling, the cell membrane signaling, always start with activation of trimeric G proteins. So when the sperm is going to bind to its receptor, is binding to EBR1, that is going to activate trimeric G protein. And when those trimeric G protein are going to activate, they are going to activate a Src kinase. In this special case, that is a tyrosine kinase activation taking place. And when that Src kinase is activated, that is going to activate the PLC gamma, which is also in the membrane. And when the PLC gamma is activated, that is going to act on PIP2, making IP3 and DAG. And IP3 is going to release calcium. And we know the first thing calcium is going to do is start the cortical granule reaction. Slow block to polyspermic. And once that is achieved, this calcium is going to bind to other calcium activated receptor, more calcium, and this PKC, which is activated together, they are going to act on sodium proton antiporter, activate them, increase alkalinity. Once that is going to happen, that is going to inactivate MAP kinase. Once MAP kinase is inactivated, you will enter the cell cycle. Okay, so the new cell division or new development is going to start. So, then we are saying if soluble factor is there, what is that? We'll talk on Monday. But that soluble factor is going to activate PLC now. Two things are going to happen. One will be the early response, one will be the late response. Once the calcium is there. The early response, cortical granule reaction, right? And whatever happened there, we said UDX1 is there, then we said that makes hydrogen peroxide, right? But then, the other important thing is you should start making new proteins, you should start making new membranes, and you should basically start the cell division. So those are the slow processes which are going to take place. And we'll go in detail on Monday. So make sure that you're at least, before leaving, you're at least sure about this part, because this will already covered.